So thanks for inviting me here today. I actually lived in Minnesota for 2 years when I was a little kid after having started elementary school in Texas. My mom from rural Arkansas, we moved up here. After 2 years, she said, "Lynn, we're going back south, with you or without you." It's probably a day like today to encourage my mom to make that statement. But it's good to experience and remember living here as a kid. So what I'm going to tell you about today is some review a few things and tell you a couple of news stories about NK cells. But I think we know why they're important based actually on work from another PhD student who was at Chapel Hill at the time, a woman named Christine Byron, who ultimately became chairman at Brown. So what Christine found in the 80s was the first human who had no NK cells but normal B and T cells. This was her New England Journal paper. And, this young woman was, pretty normal, except she couldn't handle certain infections. With the flu, she was fine. But with herpes virus infections, it ended up in the emergency room, and ultimately she died. So fast forward, this is caused by a loss of function mutation in GATA2. One copy, and you don't have NK cells. We worked on 2 patients, one from LA and one from San Francisco. They have one good allele of GATA2. They have good B and T cells. That loss of one copy of GATA2, no NK cells. I don't know why. And the problems these people have are the same. But the problems they have are herpes viruses, but also papilloma. The woman has cervical cancer. The guy has warts that can't be controlled. So, I think that's what inspired me and others in the field of NKs to focus on herpes viruses and in mouse models to figure out, what you really need NK cells for. And Tony Scalzo, some 20-odd years ago now, observed that if you wipe out NK cells, mice will die of CMV. So subsequently, we've kind of figured out why that happens. Turns out, in certain strains of mice, there is one receptor, this receptor called Li49H, that recognizes a CMV protein. These NK cells, when you get CMV infection, the dendritic cells start making the goodies NK cells like type 1 interferon and IL-15 and IL-12. That stirs the NK cells up, but they really have to have a cognate receptor to tell them where the infected cells are and how to get rid of it. They kill those cells, they make some cytokines, and they can undergo proliferation. This is the receptor complex they use, and this receptor, called Li49H, links up to an adapter protein that we found a few years ago at DNX called DAP12. I found that because it looks like CD3. It has a canonical ITAM, and it signals downstream through the same kinases as B and T cell receptor. NK cells have both ZAP70 and SYK, so you tickle Li49H, DAP12 gets phosphorylated by Src kinases, recruits SYK and ZAP, and this is augmented by a second adapter protein, another one we nicknamed DAP10. We found this because it has a YINM like CD28. This recruits VUV and PI3 kinase. This results in killing the target, making cytokines, and proliferating. Hisashi Arasi in my lab identified the ligand. The viral ligand is a protein known as M157. CMV has about 200 genes. This is gene 157. We crystallized it with Chris Garcia. Looks like class 1. This is unique to CMV. This protein exists in no other pathogen. So a few years ago, there was a student, Joe Sun, who'd trained with Mike Bevan, came to the lab and says, well, do NK cells have memory? Well, I'd read a Boole Boss's textbook, and a Boole's textbook told me that NK cells only live 2 weeks. So I said, nah, they're not supposed to, that couldn't be possible. They only live 2 weeks. We looked at the data and said, well, that hadn't really been done properly. And because Joe had trained with Mike Bevan, he'd done a lot of experiments with OT1 cells and adoptively transferred in TCR transgenics into mice and tracked them. So he used the same trick to see what do NK cells do after they've seen the pathogen. So we had mice, black 6 mice that lacked this Li49H receptor. These mice die of CMV. You put in the Li49H positive NK cells, infected, and cells took off. They clonally expanded, quite dramatically, peaked, but then the surprise was 2 months later you could still find those same cells, saying that wasn't true, that they only lived 2 weeks, because you can find them or their progeny a couple of months later. In fact, we've been able to find these cells a year later in those mice. Well, one of my contributions to the lab is I'm old enough to remember papers that are older than the students in the lab. And one was a paper by Ray Welch, who'd formally proven that NK cells actually are necessary to protect you from CMV. So Ray had taken newborn baby mice. Their NK cells aren't mature yet. They die of CMV. And Ray had shown years ago, if you take NK cells from an adult mouse and put them into the baby, you protected the baby. So I suggested to Josh Belke and Joe that they could test whether these memory NK cells were better by titrating them into the baby mice and then infecting. What they found was, as Ray had shown, if you don't give the baby mouse some NK cells, they're all dead in 2 weeks. If you put in 10,000 naive cells, again, most of those mice are dead. But if you put in the same number of these memory cells, you got living baby mice. So it looks like one of these memory cells, which has seen CMV previously, in fact, is worth about 10 of these naive NK cells. So they have undergone changes. And we could show in a plastic dish, these memory NK cells kill better, and they make more cytokines per cell. So I'm part of this ImGen consortium that's doing transcriptional analysis of every immune cell in a mouse. This is run by Christophe Benoit out of Harvard. We're the NK guys. And Ananda Golrath has been contributing RNA from various types of CD8 T cells. And what we found was that these memory NK cells, their transcriptional signature is pretty similar to the NK cell memory signature. The main difference is NK cells don't have a TCR. CD8s have a TCR. Barring that, I call them first cousins. So what we've been doing, for the subsequent number of years since we appreciated that NK cells can clonally expand and provide memory, is to ask what sort of signals do you need in order to make these long-lived memory cells. So trick that initially Joe set up and we've been using a lot subsequently, he's now a professor at Sloan Kettering, has a nice lab, is this trick of taking these Li49H deficient mice. They have everything normal NK cells have except for that one gene. You can now adoptively transfer in wild-type NK and put them into competition with NK cells from your favorite knockout animals. Put them in it 1-to-1, infect, and then ask, how do these mutant NK cells compare to the wild-type NK in the exact same environment? So this is summarizing, a lot of work, and we've done this with a number of knockouts. So if you look at what happens when you infect with CMV, the NK cells clonally expand, peak at about 7 to 10 days, undergo contraction, and then you generate some long-lived cells that are self-perpetuating. What they really need is IL-12. So if we took NK cells from a mouse that has no IL-12 receptor, those NK cells have all the right receptors, the maturation looks normal, and at steady state, they're no different than a wild-type mouse. When you infect with CMV, you get very poor expansion, no memory, and these mice are very fragile and can die of CMV infection. We also noticed that the NK cells also need to sense type 1 interferon. So when we took NK cells from a type 1 interferon receptor, again, they were pretty poor at responding. Although at steady state, you had pretty normal numbers of NK cells, mostly normal. We got intermediate levels of responses by taking NK cells that could not respond to IL-18 or could not respond to IL-33. So what happens when these NK cells see the CMV-infected cells is DAP12 sends signals, which kills the target cell, decreasing viral load. They make a lot of gamma interferon and other chemokines and goodies. In order for this signaling to work, DAP10 gives you some co-stimulation and amplifies the effect. They also need this co-stimulatory molecule we identified a number of years ago called DNAM1, which sends signals through FIN and PKC-ATA. If you take away DNAM1 or you take away DAP10, you get a really blunted response. So it gives good co-stimulation. This clonal proliferation requires IL-12 and type 1 interferon. Surprisingly, this does not require any IL-2, 15, any of the gamma cytokines. You get clonal expansion, and just as you do with T cells, that contraction, most of the cells dying off, depends upon this pro-apoptotic molecule called BIM. So if you take away BIM, Goodall and Minot in the lab showed that, in fact, the cells kind of stack up and aren't eliminated. Nice thing is that these things can self-renew, and you hit them again, and they go again. So although these NK receptors don't rearrange, they do change. So we noticed this in a lab meeting one morning where we were looking at some FACS plots of these memory NK cells that had been generated. So if you look at the expression of this receptor, this H receptor on naive NK cells, you get the normal bell-shaped curve. But what we noticed was, after they'd been infected with CMV a month later, when we overlaid, you had about 10 times more receptor than you did on the naive cells. If you do the experiment of taking the memory cells and put them into a fresh host and hit them again, they even get brighter. This only happens with this CMV receptor. If you look at another activating receptor on NK cells, like this canonical NK11, it doesn't change at all. I'm calling this an avidity maturation. So now, the NK cells that survive have the highest level of receptor and can see the lowest amount of antigen. So they have a competitive advantage to go. So these CMV memory cells, they are really good at responding to antigen. But what we noticed is that they respond poorly to exogenous cytokines, and if we give them other infections, such as listeria or the flu, in fact, they pretty much ignore it. They've learned to focus in on CMV. Goodall has done experiments of taking the memory cells versus naive cells and then challenging the mice with listeria and flu. The memory cells don't proliferate, whereas you get some proliferation cytokine-driven out of the other heterologous infections. We can explain this by if we look at the signaling through cytokine receptors. This is looking at response to IL-12, which I showed you is, absolutely essential to get these memory cells to go. If you culture them in a plastic dish with some IL-12 and look at phosphorylation of STAT4, which is the first downstream signaling event after IL-12, in fact, the naive cells, fire quite nicely, whereas the memory cells respond much less vigorously if you only give them cytokine. We saw a similar pattern with type 1 interferon. Again, the naive NK cells fire STAT1 nicely to type 1 interferon. The memory cells have a lower and blunted response. However, that's when you just give them cytokine without hitting the antigen receptor. So in these experiments, what we did was we took this tumor RMA, which we transfected with a viral ligand. If we took the memory cells and put them in a plastic dish with just IL-12, you got no gamma interferon. If we give them, however, the cognate antigen for the receptor, in fact, that triggers nicely the gamma interferon response, and it's augmented by adding the cytokine. So cytokine alone's no good, but hit them with their antigen receptor, they go. So what we learned in those studies were that these memory NK cells have increased cell surface expression of this Li49H receptor. They have an enhanced antigen-specific response to secondary challenge. They have robust proliferative expansion on a secondary or tertiary or fourth exposure to antigen. The memory NK cells are in all tissues, so if we look for where they reside, there's no special home for these memory NK cells. They're less responsive to bystander cytokines, but there is a synergy of the cytokines with the receptor activation, and they ignore other pathogens. So what do you need to get these memory NK? What we've learned is you need a TCR-like signal. You need an ITAM-based signaling mechanism. You need some co-stimulation. I like DNAM1 is probably the best one. Stimulation with antigen alone is insufficient. If we take these Li49H NK cells and just give them beads coated with M157, they do nothing. It's because they can't make any autocrine growth factors. In vivo, they need some IL-12. You neutralize that, and they don't go either. They also do need IL-15 for survival. We have taken the memory NK and parked them in a 15 knockout mouse. They're gone in 2 days, much like Steve had shown previously with the naive cells. NK cells don't produce autocrine factors. They need a little help from their friends. So we wanted to be able to look at NK cells in situ without doing this adoptive transfer. So one of my fellows who's just gone back and is assistant professor in Japan, Tsukasa Nabakuri, set up a system where we could track the fate of NK cells using a fluorescent reporter. So what he did in these experiments was made a tamoxifen-inducible Cree reporter mouse by putting this tamoxifen Cree cassette into this NCR1 or NKP46 loci, and then made back transgenics. So now, if we, for example, use some of these fluorescent reporter mice that have a yellow or a red fluor put into the ROSA 26 locus, there's flox stop codons in front of these fluorescent reporters. If we mate that by now these NCR1 Cree ERT mice, if you feed these mice tamoxifen, the mature NK cells light up either red or yellow, depending on which fluor we've used. Now, all of their progeny stay red or yellow. But you only do this when they get tamoxifen. So any new NK cells made after the tamoxifen won't be fluorescent. This worked really well. You can see here when we looked in these mice after tamoxifen feeding, we've gotten this up to about 70 or 80% now by getting the dose of tamoxifen right. But the NK cells light up. T cells do nothing because they don't express the NCR1 loci. B cells don't fluoresce. The myeloid cells also are negative. We looked at the stages of NK development of when this reporter works, and it fit perfectly of when this NKP46 gene gets turned on. If you look at the most immature precursors of NK cells, these are these cells that express the IL-2 receptor, gamma chain. These things don't light up when you feed tamoxifen. However, as soon as they start expressing the NCR1 gene, in fact, now you can see they light up, and it lights up all of the maturation stages. The NK cells are the first member of this family that's now been found called the ILCs, or innate lymphoid cells, and a few of those do pop up some NK receptors occasionally. And in fact, we do see that this NCR1 reporter works. There's a subset of these things called ILC3 that express NKP46, and in fact, that subset also lights up in this treatment. So you can use this to track those. And as you can imagine, we can also selectively knock out genes using these mice by feeding them the tamoxifen and turning Cree on in the mature cells. So Sukasa went on to ask, now I can track the fate of these cells in situ. So what he did was took some of the reporter mice, fed them some tamoxifen. At the same time, he gave them CMV. So now what you generate are yellow cells that are CMV specific. These are the Li49H positive cells, but also all the NK cells, including the Li49H negative cells, will also become colored. So you can watch them in the mice over time. And just as we had seen with the adoptive transfer, when you do this trick of feeding them tamoxifen during infection, you get expansion, contraction, generation of memory. You preferentially get the H's expanding in this process, and the ones that are H negative you see in black, they expand a little bit, but they don't have a specific receptor, so this is all just cytokine driven. We do see that both populations of cells are activated. If you use a marker such as this KLRG1, which is a really good marker for NK cells that have been previously activated and very stable, naive NK cells don't have KLRG1, but when they get activated, they really jack it up to high levels, and it stays there. Also, you see induction of granzyme B after infection, and the ones with the H receptor, in fact, do make more granzyme, but all of them get a little granzyme. So you'll remember that I told you that memory NK cells could be found everywhere, but actually the one place we found that there was a preference of finding these cells, because now you can compare in the same mouse the yellow cells without the CMV receptor with the yellow cells that had the CMV receptor. There was a bias in that you found more of the antigen-specific NK cells in the salivary gland. Now, this is important because the salivary gland is where you have persistence of CMV. This is true in humans as well as in mice. I mean, you get CMV because kids spit on each other. So that's where they hang out. And the reason is that these cells have an integrin, which is preferentially expressed on cells that like to hang out in the salivary gland, this 49D receptor. If you compare the expression of this CD49D integrin, it's higher on the memory cells than on the naive cells, and you find those accumulate in the salivary gland. Laurent Barset had published that a couple of years ago. So we also found that when we do these experiments in the mice, that the clonal expansion of these NK cells requires IL-12, because if we use a neutralizing antibody, we can really shut down a lot of the responsiveness. And that was true of the clonal expansion of the antigen-specific cells, but also the small increase in the cells that don't have the receptor. That's also driven by 12. So that is the major player. Okay. But someone asked me, well, herpes viruses, we have 8 of them. If you get a herpes virus when you are age 2, if you live to 100, you will have that same herpes virus. You never clear them. So someone kept saying, well, what about your memory NK cells? Do they need persistent antigen to keep going? So Sukasa took advantage of these reporters to answer that question, in that he took the reporter mice, hit them with CMV, gave them some tamoxifen. A month later, he isolated the yellow cells and put them into a fresh mouse. This was an uninfected rag mouse. We put them in a rag so that you wouldn't get responsiveness by B and T cells against the yellow protein. And then he could actually ask, what happens? He simultaneously introduced some naive NK cells, which had never seen CMV, and asked, well, what happens? The memory cells actually disappear. They do need antigen to persist. So we should probably call these effector memories. They're not long-term memories. These require antigen to tick over. So if you look a couple of days after transferring these cells into the mouse, which has no CMV, you can still find naive NK cells. You can still find some of the cytokine activated NK cells that had been in a CMV-infected host. But the ones that were CMV-specific, they rapidly disappeared. Well, I think that's because, they're really lousy at just responding to cytokines. And this shows you that if we take NK cells, remember that if we take NK cells, the naive ones versus the memory ones, and just put them in a plastic dish with IL-12 and IL-18 and look at gamma production, in fact, the naive NK cells go crazy. About 80-90% will make gamma. The memory NK cells are pretty lousy. So we thought, well, what does it take to keep an NK alive? From Steve's work, we know that 15 is important. So we thought perhaps these memory NK also aren't as good as competing for 15. And that ends up being true. So Sukasa took the memory NK and the naive NK. He CFAC labeled them and then put them in a plastic dish in competition with each other and titrated the amount of IL-15. What happens is the naive NK cells eat up the IL-15 and the memory cells don't get as much. I think that's what accounts for their loss. Without antigen, in fact, they really don't compete for cytokines well. Give them antigen, they win. So what we learned, from these studies was both the cytokine-activated and the antigen-specific memory NK cells can persist for 1 month after CMV. Again, we got some mice that hung around for a year. Cost me a lot of money. I didn't know they were in the colony. Both cytokine-activated and antigen-specific memory NK cells, they need 12 for generation. These preferentially locate in the salivary gland, and they depend on antigen signaling for persistence. Memory NK cells poorly compete for survival cytokines if they don't get the antigen tickle. But remember, CMV persists for life in every species. So this isn't some artificial situation. It's probably been selected for evolutionarily. This allows the CMV-specific NK cells to persist and to provide superior host defense upon reactivation. So people say, well, these NK cells against CMV, is it a one-trick pony? Can you do memory against anything else? Well, it turns out that in this Li49 family, in some mice, they could have up to 20 different Li49 genes. In black 6, they have a very similar gene called Li49D. It uses the exact same signaling machinery, the DAP10 and DAP12. And it was shown years ago by Mike Bennett and Vinay Kumar, this is an alloantigen receptor. It sees H2D. So in black 6 mice, which have H2B, they have no self-ligand. So we took advantage of this to ask, well, can we generate an alloantigen-specific memory response? So when we knew, however, that just stimulation along with antigen with CMV wasn't good enough. For example, if we take a tumor and put in M157, the NK cells reject the tumor, no question. But they don't clonally expand because there's no cytokine. They can't make their own. They can kill, they can trigger, but they can't make autocrine growth factors. They need 12 and type 1 interferon. So Sukasa set up a nice experiment where he said, okay, what I'll do is let me adoptively transfer some of these Li49D allo-specific NK cells into a host which doesn't have that receptor. And I'll give them an alloantigen challenge. And if you do that, Mike Bennett had shown these NK cells reject H2D splenocytes. There's an allo response which eliminates them. But you get no expansion and no memory. But we said, well, they need some cytokine. What happens if you give them a viral infection at the same time? So we had a CMV in which was knocked out the ligand for H. So this virus won't drive the antigen-specific response, but it'll give you all the cytokine goodies. And in fact, you get the expansion, contraction, and memory, just as we had through the H receptor. You can see here if you don't infect, but just give the allosplenocytes, in fact, kind of nothing happens. You eliminate the splenocytes, but that's all they do. If you simultaneously infect with this CMV without the H ligand, clonal expansion, contraction, memory. This is absolutely specific for Li49D. You can see that in the same mouse, we put in total NK, but only the D positive cells expand. And it requires IL-12. So again, if you do this experiment of alloantigen stimulation with viral infection to provide cytokines, if you put in a neutralizing 12 antibodies, you pretty much shut this process down. So that works. That's good. But, a lot of people will ask me about, well, what about cancer cells? Can we generate memory against cancer? Well, you've got to have a receptor a receptor signal to get the generation of these cells. So one of the receptors we know about that has ligands on cancer cells is this receptor called NKG2D. So we asked the question of, can we generate memory through this NKG2D receptor? So as you might recall, this NKG2D receptor, it's called an NK receptor, but it's actually on all NK cells, almost all gamma delta cells, all in mice, all activated T cells, as well as in humans, all CD8 T cells. It's looking for these ligands that appear on stress-induced cells. So in resting mice, this NKG2D receptor is linked up only to DAP10, which can give you these PI3 kinase and VUB signals, but it doesn't give you ITAM-based signaling. In mice, but this doesn't happen in people, you can get an alternative splicing of this NKG2D to make a spliced isoform, which now can pair with DAP12 to give you this ITAM signal. That was originally shown by Colonna and Raleigh's lab. So we ask, okay, if we actually, set this system up to provide ligand and trigger through NKG2D, can we generate some memory? So the experiment here was Sukasa mixed 3 kinds of NK cells: wild-type NK cells, DAP12 deficient NK cells. These still have NKG2D DAP10, and DAP10 negative NK cells, which, when they are activated, can turn on the isoform, which can pair with 12. Put these in NKG2D deficient mice, and now infected with a virus, which is a loss mutant. CMV is pretty good at getting rid of the ligands for NKG2D. So if we use a mutant virus, in fact, it can now express nice levels of ligands, RAY1, on the infected cells. None of this works. When we adoptively transfer any of those NK cells and look, a month later, there's no NK cells left. There's never any expansion, and they just peter away. Well, so NKG2D signaling alone is not sufficient for effector NK cell expansion or memory generation. But we ask, well, maybe, we know that, in fact, you can get the splicing of NKG2D to make an isoform that pairs with DAP12. But when we looked at the kinetics of that, it's very transient. So when we activate mouse NK cells, at day 1.5, they, in fact, make this DAP12 isoform. It's gone by day 3, so there's no persistent ITAM signaling. So the NKG2D receptor is good at triggering an effector response. Like killing, it works great. But it cannot send that ITAM-based signal to get them to differentiate or expand, even when cytokines are around. So we think that there's a lack of sustained signaling through DAP12 because it's only there transiently. And in fact, in humans, it's gone. It doesn't exist at all. So to make these good NK cells and to get clonal expansion, you need a good ITAM signal, co-stimulation, and cytokines. So when people ask me about generating memory against cancer, you got to check those 3 boxes or it ain't going to happen. I'm allergic to mice, deathly allergic to mice. I got sensitized in grad school, and that's why I worked on only human NK cells when I was at the bench for 20-odd years. We found the same comparable population of CMV responsive cells in us. We did this by Sandra Lopez-Botin in the lab. She got 50 bags of blood from the blood bank, stained them for all the NK receptors, and said, the blood bank knows who is CMV positive and negative. They gave us 50 bags of blood blind. We did all the NK receptor analysis. And it turned out we broke the code. Only people who had been infected with CMV had these NK cells expressing this receptor known as NKG2C. And they co-express this carbohydrate marker, which is a marker which comes on after human NK cells are activated, called CD57. It's the first NK marker I ever worked on, called Lu7 back at BD. I still have no idea what it's good for, and this doesn't happen in mice. So hopefully sometimes before I finish my job working on NK cells, we'll know what it's good for. But it's a great marker. So in individuals who are CMV seronegative, if you look for cells which have high expression of NKG2C and CD57, you don't find those cells. They can be up to 80% of the NK cells in CMV seropositive. Remember that this was the actual data from the blood bank. You can see we didn't need a calculator. These are normal humans walking around. They're not sitting in a plastic box. Only the people who had CMV scored of having this population of NK cells. With the transplant guys, these were the kidney transplant guys at UCSF, we hooked up with them because after transplantation, they often put the patients on they put them on antivirals because they're giving them immunosuppressive agents so they don't blow the kidney out. But some of those people, usually about 1 a month, will reactivate CMV. The docs know the exact symptoms, and they quickly, start backing off the immunosuppression, give them some antivirals. We looked at 8 patients. This is shown 4 here. In every single one of them, only the NK cells with this NKG2C took off. We did similar studies about the same time. I told Jeff Miller and his gang about these results. He did the same experiments in bone marrow transplant patients. He saw the exact same thing. This is the receptor. This is the receptor. CD94 is a heterodimer with NKG2C. My old buddy DAP12, which is the signaling unit for Life49H in mice, this is like 98% conserved in mice and humans. It does the signaling. The ligand is HLA-E, an invariant HLA molecule in humans. We're now trying to understand, there must be some alteration in HLA-E that makes this thing go. We also saw that and Jeff has seen this also, that in fact when we went back and regated in humans, T cells can also acquire this NKG2C receptor, but you only see this in CMV-infected individuals. In this person, 7% of all their T cells are co-expressing a TCR as well as NKG2C. We've done more recent work where we've used tetramers for CMV, at least for some of the common ones. And these C cells, they weren't having a TCR against CMV. So they're having a TCR against something else. We also did a very cool experiment with Christine here, studying the Minnesota students. These are the kids, they're like mice in a plastic box in the freshman dorm. She got blood from these kids during the course of their undergraduate school. They came in being CMV negative, EBV negative, or positive, and then she was able to track their EBV responsiveness during their college career. So after she published her paper in JM showing that beautiful work, I called her up and said, "You got some cells left?" Because I wanted to see if the NK cells, which are responding to CMV, would recognize EBV. They don't. In fact, using this like mice. You got them pre-infection, you got them during acute EBV infection, and then you got them up to 4 years later. They're all eating the same chow, living in the same plastic box. But it was really cool. You can see that this was true of both the T cells and the NK cells. If the kids came in CMV negative, they really didn't have any of these NKG2C positive cells in the T or the NK compartment. And that didn't matter whether they got EBV or not. So this is really a specific response to CMV. If they came in CMV positive already from having it in elementary school or nursery school or whatever, in fact, during the acute infection, we also did not see an elevation of the C positive cells during that time. So even the ones that were primed for CMV kind of ignored EBV. Unfortunately, DAP12 is never turned on in mouse T cells, unlike humans. So we wanted to mimic this, and we've done some recent studies in which we're giving mouse T cells, like I showed you that humans do have these NKG2C T cells. We're giving them Li49H and DAP12. So we've done this by making a construct in a P2A vector, having DAP12 and then this CMV receptor, Li49H. You can use this to retrovirally transduce primary mouse T cells and now give them CMV and ask, how do they work? Well, first in a plastic dish we've shown that if we take the mouse T cells, give them this CMV receptor, and in a plastic dish, stimulate them with either a tumor not having the viral ligand or a transfectant with the viral ligand. In fact, this is a cell trace violet dye. You can see that the viral ligand is inducing proliferation of the T cells, like I'd shown you happens with the NK cells. This also happens in vivo, because if we adoptively transfer these Li49H positive T cells into mice which are H negative, so there's no competition going on. In fact, and look like a week later, we can see specific expansion of the T cells expressing this NK receptor. So what's going on in the lab now is Mark Patempa is investigating what does memory look like when you drive it through a T cell receptor versus an NK receptor. What I can tell you is we do have CMV with ovalbumin in it, so it can drive a strong OT1 response. That response through TCR is much stronger. The magnitude is much higher than the NK receptor. But we're now asking qualitatively, how do they differ? And that's what I'll tell you about when I come back next time. These were the folks that have done the work, and I'd be glad to take questions. Thank you.
We'll start with questions here, but we have people in 3 other sites that
Okay.
might be interested in asking. Who would like to ask the first question or make a comment?
So do you think NK cells mainly have evolved to fight CMV, or do you think there are equivalent receptors for all the other viruses attacking humans? And I know the Li49H is pretty specific to mice, if I'm not correct.
Yes.
So, and they can have up to 20 copies. Do human NK cells have a whole array of receptors that we have yet to discover that are going to be specific for these other viruses?
I don't think so. NK cells cannot rearrange things, so you get what you're born with. And since the whole genome is pretty much sequenced now, I'll be really surprised if we're going to find some mysterious receptors show up. However, the thing is that once you prime these NK cells through CMV, their Fc receptor works much better. So now, for example, if you have an antibody response against your favorite virus, the CD16 on these memory NK cells, particularly in humans, is much more efficient. We've shown that. Several other labs, Jeff's lab has shown this. So I think that you need CMV. That was what evolved to drive this type of NK cell. But then they can take advantage of the whole B cell repertoire when you make an IgG1 class antibody against any other pathogen. That gives them the specificity. We've shown that if you trigger them through the CD16, you can get the same effector type responses that you get through the CMV receptor. But the only other virus which we have good evidence for that NK cells have probably evolved a specific recognition is in mice, is mousepox. In fact, we had made a CD94 knockout mouse, and we had it for 12 years with absolutely no phenotype. We stuck Listeria, we stuck CMV, we put in flu, nothing happened. I was visiting Louis Segal at Fox Choice at the time, and he worked on mousepox. And there was an old literature that if you deplete NK cells, mousepox is a real problem. So I sent Luis a box of all of our knockout mice, and he called me up 7 days later and said, all the CD94 mice are dead. That's the best phenotype. And the postdoc who'd made this knockout mouse and had no data for 12 years now had an immunity paper. We can't do the experiment because smallpox, which is the counterpart of mousepox, isn't around anymore. So maybe that would be the other specific pathogen.
Yep. Yeah. Lewis, Tucker, I'll give you this next. Yes. So you led right into my question a little bit. So based on what you just stated about the CD16 receptor and enhanced signaling, it should be true that cancer patients who have CMV positivity and a higher frequency of these memory NK cells should respond better to therapeutic anti-cancer antibodies that work through CD16. And the
I agree.
The second part, okay. So
I agree.
The second part of the question is, do we know if Fc receptor signaling, you looked at NKG2D and you said that wasn't good enough alone. And I think a CD16 is the NK TCR
counterpart, yeah.
Do you know if that can induce memory response the way you measured
CD16,
it?
we're doing that experiment right now. Unfortunately, in mice, ADCC sucks and they can't use zeta chain. We're fixing that.
Lewis, I wonder if you go back to your first slide.
Yeah.
The GATA2 deficient or haplo-insufficient.
Yes.
So with all of the data you've generated since then, I don't know if that was a single patient, a unique patient that no one has ever seen again, but how can you explain that level of specificity for a single gene given all of the pathways that you've described? You think they failed to develop in the marrow, or that they developed in the marrow but they were non-functional in periphery?
Yeah, we've done this. I mean, these patients are quite rare. We've had 2 of them in San Francisco that are GATA2 in haplo-sufficient, okay? And they're having, similar type of properties. We, in fact, got some bone marrow from one of these women and sent it to Jeff Miller because he's been, growing out from NK precursors, and those cells crashed and burned. So there was something wrong with the progenitors, and also the woman that we sequenced, she had a point mutation in the zinc finger of GATA2, so she had a few NK cells. We sorted them and put them in culture. They expanded for a few days and then crashed and burned. So it was a survival thing that it's providing.
So in your studies where you have the requirement for either the antigen presentation or cytokines, you need both.
You need both.
So my question is that known what the mechanism of that is? Is it a duration of a signaling pathway or an impact on the transcriptome or is expression of the receptors on the surface, those sorts of things, kind of basic?
Yeah, well, we did show, with the cytokine receptors that the downstream signaling is impaired. That's partially explained by a little lower levels, but it's not totally explained by it. So there's something in the internal wiring is also different about them. We've been trying to figure that out.
Comments or questions?
So at least, what's going on in the human field is people are trying to understand the roles of antigen-specific memory cells that are specific to CMV. And I think spurred on by some of the stuff that you've done, Lewis, people are really interested in IL-12 stimulated NK cells and saying that they're inducing a memory phenotype that is somewhat similar, some claiming virtually the same. Do you have any comments about the antigen-induced memory cells versus the cytokine-induced memory cells?
The antigen-induced ones, you're getting TCR-like signals. I mean, I think that's the fundamental difference. What we are doing right now in the signaling business is we've made chimeras with either DAP12 or zeta chain to see whether or not downstream. Both of them have ITAMs. So the question is an ITAM an ITAM, or is there something more complicated about it? So we've been getting Art Weiss to help us and made a bunch of constructs. But I think that, these ITAM things were made to give you, we know that they were made to give you T-cell receptor and B-cell receptor specific responses. So I think that's the ticket of how we're going to understand this.
So you mentioned CD3 zeta, and one of your old papers, I think you showed that CD3 epsilon can be upregulated in NK cells upon activation. I'm wondering if memory NK cells would express CD3 epsilon or any other building blocks of CD3.
In humans, they do. In mice, we haven't seen that. So human NK cells, all of them, when you activate them, epsilon gets turned on. I have no evidence that it ever gets to the surface. We looked at that pretty hard back in the day. But as Jeff mentioned, the other peculiarity is we also observed that in mice, mouse CD16 can't use zeta chain, whereas humans can. So there's these species differences that I think account for some of the biological issues. Because we tried to look for these, human memory NK cells have zeta but no Fc gamma. Mice can't do that because zeta doesn't work.
So could cytoplasmic CD3 epsilon be a marker for memory NK cells?
It's transient. So I think it's like CD69 is transient. I don't think it's a permanent change. In humans, the acquisition of CD57 looks the best. And in mice, this kind of super high expression of KLRG1 looks to be closest we get.
So I've got a question about the avidity maturation model that you had. And so the question really is there, why would they want to increase, if this is a specific receptor, why do you have this increase later on? Is there a reason why you wouldn't want to have it just expressed at high levels right from the get-go so that it could detect this infection pretty rapidly? And is it possible that this somehow is related to the non-responsiveness that these memory cells have later on? Is that linked to this avidity maturation, or is that independent?
I think it's, there is plenty of receptor obviously there. You can see, by fax, there's already several thousand copies. But I think what it does is, if as you have lower and lower amounts of antigen, the ones with the highest amount of receptor just get Darwinianly selected out. And I think what Joe Sutton, one of his students, has actually done some experiments that are compatible with that interpretation.
Anyone else? Thank you.